The CNVScope app takes a whole genome approach to visualizing copy number data sets through the calculation of an all versus all linear relationship matrix across the genome, beginning with copy number data taken from the Genomic Data Commons archive. And the package is configured to take either TCGA or target formats. All of this is detailed in the vignettes, which is available within the package, installable from GitHub, and also advertised on CRAN as well. CNVScope does not just display copy number, like this plot in CNVKit, with chromosome position along the x-axis from chromosome 1 to the sex chromosomes at the end, and then copy ratio along the y-axis. It also doesn't display unidimensional single position peaks like Gistic does. Rather, it shows interactions, and it shows interactions in a way that is more easily interpretable than network diagrams. This is actually from CNVScope documentation, and it shows them in a way that's more interpretable than network diagrams, which can become cluttered with too many interactions. We have 8 million different interactions we're mapping across the interactome, and it becomes difficult to see it in the same way that you can see domains and peaks in our visualization tool. Similarly, I'm going to show you an example of standard copy number plot along the bottom, and then standard CNVScope plots. This is for the whole genome, and this is for chromosome 5q, the Q arm of chromosome 5. And you'll see that it's really hard to tell what's going on in that mess, really. As a final example, there's a journal that is aptly titled From Hairballs to Hypotheses. Complicated network diagrams are sometimes aptly named this way because it's difficult to understand what's really going on. In contrast, CNVScope takes a whole a two-dimensional approach like Hi-C does. And if we zoom in, not that far. And we see chromosome 1, 2, 3, 4 through the sex chromosomes on either end. We can see exactly where they start and end. And we relate using this plot instead of Hi-C, where we're relating physical interactions such as looping events, like we'd see in Juicebox or other common Hi-C tools. We are actually having copy number relationships mapped across the interactome. And we relate copy number levels in every location to every other location across the genome using metrics based on linear relationships. Darker regions reflect stronger relationships. Red reflects positive association. Blue represents negative association. White represents little to no association. For your information, the metric used is the sine of the correlation multiplied by the negative natural logarithm of the p-value for the test that the linear slope coefficient beta is non-zero. This is we will be able to see the linear regression line and the observations plotted that gave rise to this metric for each individual point shortly. Note that the strongest signal actually appears along the diagonal with large blocks denoting chromosome arms. The fact that these signals appear as large blocks reflects the tendency of chromosome arms to have the same copy number across the entire arm, but not always. This is chromosome 1 that exhibits this behavior. This is chromosome 2 that does not. This makes sense, as the P and Q arms often behave independently in copy number data. Reduction in the immediate off-diagonal signal between the largest blocks, between this block and this block right here, there's a reduction in signal, corresponds to the position of centromeres, as frequent rearrangements at centromeres result in reduced off-diagonal correlation near the centromere. In our paper, we detail a region in the vicinity of NBAS and NMIC loci that had recurrent rearrangements in clinical cases, sometimes associated with NMIC amplification. We wish to show that this important region has a plainly visible pattern in CNVScope. And what you can do is you can select the chromosomes, you can select the saturation limit. This stops high values from drowning out your signal. It's good to keep it at 75 for this particular plot. 1024 is also a good resolution to keep the height of the heat map at. So the other thing to do is you can also do a gene search, and it'll find the actual ensemble 75 coordinates. Saturation limit, to talk a little bit more about that, if you turn it up, it'll make the plot a bit dimmer, and it will lowering it will make the plot more colorful in very simple terms. But basically it will it sets the ceiling for the scaling function. If you want to see it, I've documented it fully for those technical folks out there. And that's in the package and in the RHelp. So if you look in the plot, you can find regions that are negatively associated, and over here as well, and you can see the genes and the values at each individual location. Higher values designate larger relationships. They've been scaled such that the values near 1 have a very, very strong linear relationship. The values near 0.5 have nearly no relationship, and the values below 0.5 have a strong negative relationship the closer they get towards 0. So what's obvious is there's a number of things. One, we have domains along the diagonal, more than 2, as we talked about earlier, and there. Additionally, we find this cross, this place where there is no red domain where there is a strong linear correlation between the copy numbers. And if you zoom in right in there, you'll find what you're looking for. You'll find NMIC right along the diagonal, and next to it, NBAS. So they're very, very close to each other, and so it makes sense that rearrangements that would affect one are closely related to the other. And they're definitely in the same region. So there's that, and you can zoom out. You can also reset the axes back to the default view. Now, if we want to get some information on a specific gene in the region, we can click anywhere, and we'll get the genes that happen to be there. Probably the best place to look is in NMIC, because we know that's a cancer census gene, and we'll see that right here. You can search for things and filter down using these search boxes. Sometimes you'll get a lot of genes in certain regions, and that's what that's helpful for. You can also increase the number of entries by quite a bit if you like, whatever works for you. There's also this mini map. If you scroll over, it gives a 10 by 10 view if you want a closer view as well. So many options to explore the interactome. So the next thing we'll look at is the cancer gene census data. This is straight from cancer gene census, and it tells you what specific tissue this gene has been found in for cancer, and it also gives its role, its precise location, which we already know, but it also gives translocation partners that are known, other syndromes, and other names associated with it. And it designates whether this was found from row or column loci. Moving on. It also has a search box if you get a lot of cancer genes in a certain region, just in case. Now, if you look at sample level information, if you're looking along the diagonal, you'll get great correlations, great relationships, and everything. But if you go a little bit off diagonal, you'll find that things are a little more different. If you go over here, for instance. This case, this is a strong positive relationship. There are some that are high over here. This gives the samples they come from. This is actually a good example. It gives clusters of signals and where they happen to be, and what precisely what the precise values and samples happen to be for those values. And so you kind of see a very granular view of, for each bin pair, what are the individual samples and what are the individual copy number values that gave rise to this particular calculation. You can see it. You can see the precise location. For instance, here we have a value of 6.58 and a value of 1.01 for copy number. And you can actually zoom on this particular graph too. And you can see individual ones more clearly if you like. And it's all right there for you. You can reset the axes. You can also adjust the opacity of this particular plot in case you want to more clearly see what color these things are, or see through them, whatever works. Further, lastly, you can see the expression values. Now you see the most variable ones given by this row variability percentile. Find that there are a lot of different kinds of genes. There's miRNAs, link RNAs, snRNAs, but the protein coding gene that happens to be most variable in this region is C2OR43. There are also something a little more, with a bit more conventional of a name would be TRIB2. We'll also find, you can also sort by the row mean. You'll find that this particular region, there aren't a lot of genes in this region. I picked it so that you'd see something different than something along the diagonal, but you are able to find what genes are expressed in that specific location. So I've kind of given and if you want to go back to the whole genome view, it's right here for you, and it will let you adjust the saturation. If you want to zoom in or zoom out, I'd recommend using Chrome. That way you can zoom in and see precisely where each chromosome begins. We wanted to show you that this is chromosome X over here, and this is chromosome 1. You can see precisely where chromosome 1 starts and ends, and then you can also see chromosome 2, where it starts and ends, and then you can also see chromosome 3, 4, 5, 6, 7, 8, 9, 10, and you can see off diagonal regions that may be interesting to look into. Feel free to also take a look at the package. We have a great set of installation instructions and show precisely how things work. And